welcome to Sano Seminar. So, well, now we can slowly start the seminar, which is the last one before Christmas and last one in this year. And we are very happy to host Dr. Ekaterina Kutafina, who is the leader of the team Biosignals from Institute of Medical Informatics at the Aachen University. Aachen is in Polish Aquisgrad, and it is very close to the place where our partners from teaming are having their place, namely close to Illich. So from some point of view, you can consider that it is just a number of Illich. Ekaterina has completed studies in mathematics, and he obtained a PhD degree from HGH University of Science and Technology in Krakow in mathematics. And after a few years after that, she also obtained a PhD degree in Germany on computational medicine. And so in her group, she is closely in collaboration with hospitals, and these research are related with processing visual data. And today, biosignals will be the main topic of her talk. So, Ekaterina, floor is yours.
Okay, so thank you very much, Marian, for the introduction. I'm really happy to participate in Sano's seminar. I also very much appreciate the raise of such a computational medicine institution, particularly in Krakow, because that's a city I feel very much connected to. I have lived in Krakow for 20 years. And as Marian said, I accomplished my first PhD at HGH University, which I'm still affiliated with. So today my goal is to talk a little bit about the topic of my current team, which is called Biosignals, and about general perspective and challenges of biosignals in computational medicine. Okay. So the outline of the talk would be as following. So first of all, I'd like to talk a little bit about where my team is in the landscape of the university. And then I will talk about several of my key projects, and I will spend most time on this first project, which is called Digital C-Fiber. It's a project on neuropathic pain. And then just briefly talk about some of my EEG projects and one project on the detection of epileptic seizures, which is called Monikert. And then I'll spend the last couple of minutes on discussing the perspective and challenges which are related to biosignal as promised. Okay, so starting with the institutional context, my team is the part of University Hospital of RWTH Aachen, which is one of the largest technical university in Aachen with actually great computer science faculty. And the Uniclinic is simultaneously a separate institution, but it's also faculty number 10 of the university. The number of employees in the clinic is kind of comparable with the number of employees in the rest of the university. However, of course, the scientific staff percentage is lower at the clinic because they have a lot of just purely hospital staff. Okay, and within the hospital, we have a department of medical informatics. And now what actually is medical informatics? There are many definitions, and the one which I kind of like is which is placing medical informatics as a part of general health informatics. So this definition says that it's about collection and evaluation of medical knowledge and patient data to facilitate and improve patient care. So the definition sounds quite general, but basically in these units of medical informatics, we typically doing some work which is in particular related with infrastructure for research and medical data. And another popular topic is any sort of artificial intelligence related technologies, decision support systems, and so on. Right, so in Germany, medical informatics is at the moment quite a big thing. So there exists a large German initiative of medical informatics, which contains many consortia within it. And I quite like this map because it's also showing the institutes of medical informatics across Germany. So you can see here Aachen, which is really pretty much on the Dutch border. And Aachen is a part of the consortium, which is called SMITH. And one important part of SMITH is building data integration centers. So those centers are aiming in actually giving a possibility to use hospital-generated data for research, which is quite important because it's a large amount of data, which usually researchers have a lot of difficulties to get to, in particular because of multiple legal issues related to patient data. So within the Department of Medical Informatics, we have several divisions. We in particular have research infrastructure, education in medical informatics, medical software engineering, data management software engineering, and my own team, which is called Biosignals. Now, how do we actually define biosignals? So again, as with medical informatics, you can find many definitions. I like to define biosignals as time-varying measurements of, let's say, human physiological activity. So the time-varying compound is the most important thing here. So typically when we're thinking about biosignals, we have in mind electrophysiology recordings such as electroencephalography, electrocardiography, and so on, which basically means that we are getting several thousand data points per second, which is really very dense in time data. But we also can think about measurements such as daily glucose measurement. If we're doing it across time, even if it's a relatively discrete measurement, we keep repeating it, we're still getting in a long run, we're still getting some sort of time series. And particularly in combination with other biosignals, they also could be treated as a type of biosignal. So this time series component is one of the most important. And the long-term goal of my group, I would define as integration of different modalities of biosignals to better understand human physiology as a highly interconnected network of different individual mechanisms. What does it mean in practice? In practice, it means that we're working with many different types of biosignals. We have many separate projects, but we always have this large goal in mind about data integration. So I think with the current availability of the data, we should be able to get to the next level of knowledge by treating human physiology not as many separate systems, but instead of this interconnected network of different physiological systems within human body. Okay. So now typical modus operandi of my group is as here on the diagram. So we're getting data. My cursor is visible. We're getting data typically from different sorts of sensors, in particular electrophysiological sensors, from which we're extracting those biosignals. And then we're building data-driven models, which basically means that we are not usually assuming any specific rules which are governing the data. Instead, we are basing our knowledge purely on the data. And we're using lots of machine learning to build those models. And the center of our interest is personalized medicine, which means that we are aiming into improving a personal outcome for the patients. So in standard medicine, we usually think about patients as a certain kind of statistical population, but what is actually statistically the most beneficial for the group of patients is not necessarily the best outcome for very specific patients, right? So in personalized medicine, we are trying to focus on the specific characteristic of a given patient. So about in a little bit more details. So typically we're dealing with this kind of problems. We're working a lot with mental health issues. So we have a group of patients, for example, with schizophrenia, with depression. We are working very closely with the Department of Epileptology in our clinic. So seizure detection, we are working on chronic pain issue, particularly with neuropathic pain, and also with patients who have diabetes, but also in terms of damage of peripheral nerves. So that's also related to chronic pain. The data which we are typically getting is either electrophysiology, which is again electroencephalography, EMG, it's the electrical muscle activity data, or ECG, so heart activity data. We like working a lot with wearable sensors, so anything like smartwatches or wearable ECG patches. This is of a lot of interest for the team. And we're also getting some sort of supplementary data, which is not directly electrophysiology. So for example, data from electronic diaries, which can serve as some sort of baseline knowledge for the recorded electrophysiological signals. The methods which we are using are pretty heavy in signal processing. And then, of course, we are using different sorts of machine learning algorithms, including deep learning. We are also using computational simulations. So in particular, we're using programs such as Neuron for simulating single nerve activity to compare it with data which we're obtaining in the experiment. And we also performing quite a lot of spike analysis for also single neuron data. And we're using many methods for data integration. So in particular, building all kind of systems to support, for example, metadata storing and general organization of the data, so that we can integrate the data from different experiments from different groups into one research. And a typical output of our projects, outputs of our projects are most importantly, I would say, algorithmization of the problem, because what often is happening is that we're talking to medical professionals and they have some sort of medical problems which is not directly put in computational terms. So after this discussion, we need to try to break this problem into smaller pieces which are possible to algorithmize and translate to the terms which could be handled by using mathematical modeling. We're supporting also medical professionals in getting the data into some sort of usable form, because this is particularly an issue with Biosignals data. It's could be very noisy. There is often some metadata which is stored in paper journals, lab journals. So we need to support making the organization of this data of good quality for further research. And further outputs which we are aiming at are decision support systems. So based on, for example, machine learning algorithms, we would like to support clinical decisions. We are trying to build diagnostic tools and also research tools. Okay. So let me now tell you something about my, at the moment, main project, which is taking about at least 60 to 70% of overall research time. This project is called Digital C-Fiber. And the medical problem behind it is painful neuropathy. So painful neuropathy is a kind of a problem of peripheral neural system with neuropathic pain in the foot as one of the most disturbing symptoms. So this kind of pain could be related, for example, for damage due to diabetes or due to some mutation in, for example, sodium channel. The research is linking this kind of neuropathic pain in the extremities to spontaneous firing in so-called synaptoreceptors. So those synaptoreceptors are neural fibers which are responsible for different sort of perception, peripheral perception. And the need for this medical topic is to develop diagnostic tools, which are virtually non-existent at the moment. Any sort of objective markers for neuropathy are really non-existent. The only thing which is being used is the pain scale. Also, really targeted pharmaceutical solutions are pretty much in the also very basic state. So in order to support the development of this objective biomarkers and pharmaceutical solutions, we need to understand the biophysics behind the pain. So what we really need to understand is pain encoding through firing patterns on those C fibers. And we are collaborating with Barbara Namer's lab on micronography, which is also part of the Uniclinic in Aachen. And micronography is a very rare technique, which is simultaneously the single available method to record from awake human subject C fibers. Right. So as you can see on the figure, what's happening, we can put very, very thin needles into a bundle of human C fibers. And then we can also produce some sort of a stimulation on the foot. Here on the figure, you can see mechanical stimulation, so you can just press the finger in a sensitive area. But we can also think about other stimulations, such as electrical stimulation, chemical stimulation to induce itch, for instance, or heat stimulation. So with micronography, we can record both resting state activity of C fibers, but also evoked activity with this stimulation, which I just mentioned. Okay. Now, it seems like, okay, we can get an access to the signal from those C fibers, we can do certain stimulations, and that's a straight way to analyze the pain signaling. But the problem here is that we typically put this needle not in the single fiber, but in a bundle of fibers. So we're getting several signals which are overlapping, and actually just one source of recording. So with multi-electrodes, it's easier to separate those fibers, but we only have one electrode. And putting multi-electrodes in this fiber could be damaging for the nerve. Therefore, it's not used, at least with the current technology. Right? So we have to figure out how from this single signal, how we can possibly separate the fibers. So historically, in micronography, so-called marking method is used to figure out what's going on with those fibers. So what is done is that we're introducing every regular time interval, let's say every 4 seconds, we're introducing electrical stimulation, which is marked here as this blue spike. That says electrical stimulation. And on this figure, you can see this hypothetical situation that we have 2 fibers, main fibers in our recording, right? So we stimulate, and then we see the response of the first fiber, and then we see the response of the second fiber. Because typically what's happening is each fiber has a little bit different propagation speed. And normally speaking, we wouldn't notice maybe that there are 2 different fibers, but if we keep repeating the stimulation, what we see is more or less consistent in time response. I'm sorry, there's any question? Jan, I see the hand
yes,
raised.
there was a question in the Q&A. I'm not sure if you can see, I can read it. So it's from Aneta Lisowska. Yeah. Psychological and perceived pain may differ. Do you consider both for decision about the pain medication, or first focus on the physiological objective pain? So
okay, so first of all, I'm not medical doctor, right? So I'm not the one who is deciding about treatment. I'm only supporting data analysis here, right? So yes, of course, so the pain is the thing which is happening in your brain, really, not in your foot, definitely. And pain is basically really a thing of how you're perceiving it in the brain. So I'm not interested really in pain as such. What I'm interested in how the certain stimulation is encoded. So if there is stimulation which should be painful, for example, when it's a certain kind of certain level of heat and so on, we're just tracking the signaling on the nerve. And we also see very different responses. For example, we have some patients with lower thresholds of pain, but we can also see it on encoding on the single nerve. But of course, yeah, it's absolutely it's absolutely different things, pure encoding in the nerve and brain response. But I'm not sure if it's actually answering the question.
Well, I can enable you to speak.
Okay.
Yeah, thank you. Okay.
Okay.
So
thank you. Right. So right where we were. Okay, so we're introducing this marking method when we do this stimulation in regular intervals, right? And then we're able to track, due to this consistent response in time, that we have there is one fiber which is consistently responding after a certain time, and then there is a second fiber over here, right? So this way, the specialists on micronography are able to figure out how many main fibers they have in the recording at all. And then they also vary the stimulation in the way that they can assess the types of these fibers, because C fibers are also different between each other. They respond to different kind of stimulation. Some are, for example, responsive to mechanical stimulus, some are not, and so on. Now, what's happening if we introduce additional double pulse? The first fiber responds to the double pulse. After some delay, the second fiber responds. But then those responses to our regular pulse are both a little bit delayed. And this is called activity-related slowings. So if there is some activity on the fiber, the speed of propagation of information on the fiber is a little bit slowed down. Now, the same holds if we have neuropathic firing. So typically, if the fiber had some neuropathic firing, which is related to pain, typically we also observe some sort of a delay. So one of our main projects is to figure out how the size of this delay and the recovery function after the delay can help us to figure out on which fiber this neuropathic firing happened. So we're trying to build some sort of probabilistic model to support full fiber separation. Okay, but in principle, the whole work, until we will be able to analyze pain encoding, contains many pieces which need to be put together. So we're getting this data from, we're also getting additional animal experiment data. We have this healthy human volunteer data, and we have data from patients with different sort of pathologies. And one of our most important work packages in this whole story is data organization and unification, right? Because we are getting all sorts of different formats, also from different groups which we're collaborating with, different kind of protocols, different people who are performing the experiments, which have different ways of doing stuff and describing stuff. So our main actually work, the most time-consuming work, is to organize and unify this data. And then we are working on this fiber separation. We are doing it through analyzing how the spike activity is related to this activity-dependent slowings, which I just showed you before. And then the future steps would be to actually find those unevoked latencies, which are related to neuropathic pain, and quantify those spike trains and compare the spike trains between patients and healthy volunteers data. Right? We're also working at the moment on the spike trains because we have some data sets which have relatively clean situation. For example, we have one large fiber which was managed to record, and therefore we can already start working on the spike train before we solve the whole problem of the full fiber separation. And we're also building a special software which is called Openmnglab, which is helping us to go through all this different work packages in steps. So here you can see, okay, so we have different type of subjects, we have different type of stimulation, and we're working typically with those three types of software, which is DAPSA, Spike2, and Openmnglab. In Aachen, we're using DAPSA and Spike2. Our partners in Bristol lab are using Spike2 and Openmnglab. And Openmnglab, so our software, is a modular software, which is in the first step supporting data unification from those different formats. We are doing it using a Python-based format for electrophysiology recording, which is called NEO, and also ODML tables, which is a tool to support storing metadata for electrophysiological recordings. We also have some modules for pre-processing of the data. So for example, wavelet-based filtering. Here is the part which is supposed to sort the fibers. So we are able to do some fiber sorting for relatively simple data. We're still struggling quite a lot with human data. And we also have modules for spike train analysis. Once enough data is collected to have enough statistical power, those spike train quantifiers could be further statistically analyzed. Okay, so this is the main story behind our neuropathic pain projects. I'm actually very happy to be a part of this micronography research because it's very rare technique. So not much computational work is done in this area, and I'm sure that there is a lot of really interesting potential in those projects. Okay, now a bit shorter stories on some other projects. We have, for example, quite some projects on EEG research. It's related to electroencephalography. In particular, we just recently finished this project, but probably it would have some further continuation. So we worked with this device, which is called emotive epoch. This is, let's say, mobile EEG device, which was developed not for clinical purposes, but actually for computer gaming. You can quite easily mount it on your head because it does not require the special glue for electrodes, but just a little bit of saline. And so we have built up the system which allows to synchronize this EEG device with a presentation of the stimulation on the just regular tablet. And what we were interested in is mental load assessment. So for example, patients with stroke or some psychiatric patients, they are doing cognitive trainings, which means that they are training a certain kind of task. They're doing it on the computers, but it's quite difficult to figure out what is the overall progress of this training in more general terms, so not just performance of this specific type of training. And therefore, we could use some standardized psychological panels which have several difficulty levels, right? And then we can present those stimulations, and then you have to react to those stimulation by tapping on the screen. And then we can simultaneously also record the brain activity data. So what we had is two psychological panels, which are called NBAC and Go, No-Go tasks. And they both had 3 difficulty levels, plus we had a baseline difficulty level when the subject just have to stare at the cross on the screen, so basically doing nothing, or at least trying to do nothing. And we achieved the results up to 86% recognition, considering that it's between 4 groups. That was quite good. And we were using on purpose only very robust features from EEGs, which are frequency-based features, so the standard alpha, beta, gamma, and so on powers. What we discovered, and what was also not very surprising, if we just train our models on the data from some patients and test it on the new patients, sometimes we are successful and sometimes we are not. And it's working much better if we have a little bit of data from the specific patient to additionally personally calibrate the model. So in this case, it's working much better. Okay, and then the other part of the project related to electroencephalography is the projects which are based on publicly available data. I very much like this database from Templeton University Hospital, which contains more than 20,000 EEG records with a little bit also of medical information available, such as some comments from the physicians and some information on medication. So it's not fully reliable information. You cannot treat it as a full-blown clinical study, but you can learn a lot about the feasibility of some hypotheses for the potential clinical study. So what we are trying to learn from this database is how different sort of psychotropic drugs or other drugs used in psychiatry are influencing EEG power bands. We're getting quite a lot of interesting results. However, for example, in this kind of study, we cannot control the dosage of the drug. but we can have a general idea what type of drugs are doing what based on really lots and lots of records, which you typically wouldn't be able to get in a single clinical study. Yes.
Okay, we have one more question from Jacek Łukopolski, which is actual speed of the nerve pain signal as measured in your research. So if you comment on that.
Like, well, it's actually, those kind of questions are always tricky for me because I never think about, you know, the subjective units. So, let me just try to think about it quickly. So we have the nerve about, well, we can think roughly speaking 10 centimeters length, right? And we're getting something like perhaps 12 milliseconds maybe of the delay, right? So that should give you some sort of an idea. Right. But those fibers are relatively slow because they're unmyelinated, which means that the actually practical propagation speed is relatively low. Okay, not sure if that answers the question, but yeah. Okay. Oops. Okay, so the last project which I would like to mention is Monikert, which is the only projects of the mentioned which I haven't started myself. So Monikert is an existing startup which has a proprietary ECG patch for detection of tonic seizures. So that's the Monikert is a project between this kind of privately funded startup and our epileptology department and some other partner epileptology departments in Germany. And the idea is that this ECG patch is put on patients with epilepsy, and then the ECG data is collected. Then we extract R2R peaks to analyze heart rate variability from this data. And we have a quite large data collection of more than 70,000 hours recorded together, most of them with also EEG data. And this data is annotated, so whenever a seizure happens, there is a label, right? So at the moment, Monikert already has developed deep learning algorithms for detection of the seizures. And the current situation is like we have a certain subgroup of patients, which is quite large, where the seizures can be detected with high sensitivity and specificity, and another group of patients which for some reason the data somehow is different and there is a large problem to detect the seizures. So my goal now in the project is overtake further development of those algorithms and try to personalize those algorithms so we can either improve the results for this non-responsive group of patients, or at least to find out why exactly this is happening and what is different about the data that it's not reacting to this kind of algorithms. Okay, and that would, yeah, sorry. Yeah, there's a question.
I think it's a comment, but maybe
Okay,
I can enable
okay, that's after,
speak,
yeah. I guess that's about the speed.
But I think it's about the
nerve.
speed, yeah. So,
yeah.
Okay.
That kind of probably makes sense. Yeah. Okay. Right. So what I would like to do in the last minutes of my talk is to discuss a little bit the perspective and challenging of biosignals. And perspectives are kind of from where I sit are pretty simple. So first of all, old-fashioned biosignals as used in the clinic, they are very useful. EEGs, ECGs, they're gonna stay with us forever due to the high time resolution they have. Really, they're giving us really, really nice idea about different aspects of physiology of human body, right? But since we are researchers, we are interested in, of course, in the progress, and I would divide the perspectives in two different types. So one type is old sensors, new methods, the other type is new sensors, new methods. right? So all sensors, new methods is basically the what's happening right now with existing clinical sensors due to the development of digitalization and development of computational methods. So for example, we are taking good old-fashioned ECG data or EEG data, and we are now with the new methods of analysis, we are able to perform some sort of automatic decision support. So for example, do automatic abnormalities detection. It's at the current stage, it's not working anywhere close to perfect, but it's still very helpful to, even if both sensitivity and specificities are low, it still helps the medical doctors enormously. The other thing is that we can now try to integrate the data from different units and, for example, compare different subgroups of patients or even within the same subgroup of patients and get qualitatively new information from this kind of data integration. But it, of course, requires this kind of intra-hospital data research management solutions, which is in practice quite difficult to achieve. And also it requires some legal solutions such as agreements of the patients, of course, to share the everyday hospital data. That's not clinical study, that's just everyday hospital life. And the second part is new sensors, which are typically by definition going also with the new methodologies. There is at the moment, as you know, a big development in the cheap wearable sensors, right? So this kind of EEG devices, they are not super cheap, but in principle, people can afford to buy such a device and learn something about their own brain. People definitely can afford to get this kind of devices for heart rate tracking, right? Those ECG patches, they are also relatively low cost. Personally, I'm a really big fan of those devices for continuous glucose monitoring, right? So it's improved patients' life enormously when you don't have to draw your blood with a needle several times per day. Moreover, those devices for continuous glucose tracking, they are not only used by diabetic patients, but they're also used by athletes or just people who are interested in their own physiology in order to track their metabolism. So it turns out that different people are reacting to the same kind of food in completely different ways. So some people are getting glucose spikes after eating bananas, and some after eating cookies, some after eating oatmeal. So it's not exactly how it was thought of before. So it really allows people to learn a lot about their own bodies. So increasing personal awareness, I would say that it's a really big advantage of the sensor development and biosignals. And then, of course, those kind of devices, they are helping to boost telemedicine, because not only we can now talk to a healthcare provider, but we also can do reasonably good quality measurements also completely in a home environment. Moreover, those kind of sensors often allow to record 24/7, not obviously the ECG device, but for example, those kind of ECG patches definitely enable to record 24/7. With this data, we can also support different kind of clinical intervention and decisions, like, for example, decisions related to cardio patients or depression patients or patients with epilepsy, right? We can see that if on certain drugs we have more seizures, we perhaps would like to change the dosage of this drug. So it's a lot of perspectives for those kind of wearable sensors, which I can see. But now, of course, this development is coming with the challenges. And challenge number one, which I don't want to discuss in details, but it's always would remain challenge number one, is ethical and legal issues. So basically, we can think about it as a part of general artificial intelligence / big data discussion with some medical twist. I quite enjoyed this book called The Circle, which is specifically focused on the signals collected with the smartwatches and similar devices. So basically exactly biosignals. I highly recommend this book to get an idea about the ethical and legal challenges. However, today I would like to mention the challenges which I consider to be my kind of personal quest. And those are three types. So first is fair and interpretable computational methods in biosignals. Second is supporting transdisciplinary research. And the third is physiology as a holistic network. So this third one is quite a big topic, which I would prefer not to focus today. And instead, I will just tell you some more about those two first points. Okay. So I'm sure that most of the participants had some sort of experience with FAIR guiding principles. I think one of the first papers is this one from 2016. So FAIR basically means findable, accessible, interoperable, and reusable. And those are the guiding principle of how we should handle scientific data in general and medical data in particular. So for my biosignals, it is extremely important because biosignals are heavy data, complex data. We're doing a lot of stuff with this data. So this data undergoes many changes. Like, for example, we have raw data, and then there is some pre-processed clean data. Then we're extracting some features. It's the next level of data. So it's extremely important for my group to control all the steps of data analysis according to these FAIR principles. What we also often see in practice is that different labs, when they're recording data, this metadata is stored in paper form. A lot of metadata is basically stored in the memory of the experimenters. So we need to support and help to develop the tools to actually describe the data and store all the relevant information in the way that we can find it, even within the same group when the people are changing, but also so we can share this information across different kind of groups. It's also, for ethical reasons, super important to think about reusability of the data, because we're asking patients or controls, we're asking volunteers to donate their data to science, and we don't want to ask them to do it several times if we can reuse the data to answer several different questions, right? So it's also very important that we are trying to reuse the data whenever possible. And that's one of the reasons that we're building this Openmnglab, so we can handle the data in this according to this FAIR guiding principles. The second thing here is interpretability of our machine learning methods. So I guess many of the participants are using in some ways machine learning. Machine learning is great, particularly when we have some sort of solutions which we wouldn't know how to handle otherwise. And sometimes machine learning is just enables us to drop our data in the black box and get something usable with. But it's extremely important to try to track what do we have in this box as much as possible, right? So I very much recommend this book on interpretable machine learning, which allows to kind of analyze what is it we're putting in our machine learning in the simplest version we can think of, analyzing the features which we're using to plug in machine learning to analyze how important individual features are. And this is not easy task to do, but it's possible. Point: fair data important, interpretability is super important. And my personal favorite, simplicity. So what I would like to comment here, and I guess also a lot of participants possibly have experience with this kind of things, when we're applying for different fundings, what reviewers want to see is cutting edge machine learning methods, which are not always what the medicine really needs. I mean, cutting edge, great, but we really need to control the benefit of those fancy methods, right? So what we did in our pretty recent paper, we took our own pipeline on automatic detection of abnormalities in EEG signal. It was not very complex approach, but we had some shallow artificial neural networks, and then we had some voting system, some pre-processing before this, so it was quite some step. So what we did, we just kind of disassembled it in separate pieces and try to see, and try to see what is the benefit of this, of this adding the complexity of the project. So it turned out that our full setup was giving a bit above 80%, but in the end, when we went to adjust a single wavelet-based feature and thresholded it, we got 75%. Of course, and now there are publications available with very complex deep learning models, combinational, convolutional, recurrent networks, and so on and so forth, which are giving another 5%. In the end, you're ending up with something like 86. So of course, for the purpose of practical use, this 10% matter. But we need to realize that one single feature gives us 75, right? So I think it's really quite important. And maybe if we just work a little bit more on it, we could just get a couple of features which are giving, a very simple percentage, and we also understand what they're doing, So I think it's very important to track when this complex algorithms are really necessary and what we can achieve by using simpler methods and kind of compare and have at least baseline to know what's going on this simple version. Okay, why it's not working? Okay, and almost finishing. Okay, so another thing is this supporting transdisciplinary research. So we typically have some data, some preparation, and some analysis in the end, right? And in classical hospital data, we are getting some sort of tables. They could be huge tables, but they're still tables. And then they are going into some sort of statistical analysis or even some sort of machine learning if it's enough data. And that could be done by basically a medical doctor talking to a scientific programmer or computer scientist, right? But with biosignals, but also with medical imaging, with anything which is remotely more complex than a table, we are getting to the situation when we have some sort of complex pipeline which needs to be performed before we can go from this data to the actual analysis, right? And in this case, it's often impossible for medical doctors and computer scientists to talk to each other. So we need someone in between, some sort of a translational researcher, which can understand many aspects of research. So in particular, some parts of medical research, some parts of this computational research, possibly software engineering, and so on and so forth, right? And at the moment, the ways to support, and it's more than just interdisciplinary. It's really transdisciplinary, so we need knowledge on this, deeper knowledge on those many aspects, so we can talk to each other. And we can do it through transdisciplinary education. And what I see in practice is that when we have students working in this kind of interdisciplinary research, what's happening, they're getting graded not very good by both faculties, because, for example, the thesis is not good enough for computer science. It's also not good enough for theoretical medicine, but it's practically very useful, right? So we need to develop some sort of way of talking between the faculties. So also we can support, and the students are afraid to take those kind of works often. So we need to talk and to figure out the ways that, for example, such a work can be graded in the way which it deserves, really. We also need to give the students the truly transdisciplinary experience, which means that we need actually supervisors who are able to supervise those kind of projects. And the other thing is that we often, as most of us have some computational backgrounds, we're often thinking about giving computational professionals some additional medical training to learn about medicine. But what we also should think about is giving the medical professionals some computational training, because it's actually many young doctors who have some sort of idea about programming. They don't need to do any very complex programming, but if it's enough if they understand some basics of what they can ask of their computational partners, that's perfectly sufficient. And that's this kind of training we should also support. Okay, so very roughly conclusions: wearable sensors and the data-driven method on all sensors can bring really, really interesting development and possibly a qualitative jump on human physiology. But we need to support building good clinical and research data infrastructure. We need to keep in mind the fair data principles, interpretable and as much as possible minimalistic methods, which also maybe could be interpretable in terms of what doctors think is interpretable method. And we need to give our full support for truly transdisciplinary training. And thank you very much for your attention. I wish you also great holidays.
Many thanks, Ekaterina. Many thanks for this talk on biosignals, and especially for what was in the conclusion.